What if the key to slowing down aging isn't found in a new pill or a surgical procedure, but in a molecular dial that can tune our very genes? Imagine a technology so precise that it doesn't just cut DNA like a scalab, but actually adjusts the volume of our genetic expression, turning down the noise of cellular decay while keeping the music of life playing. This is no longer science fiction. It is the emerging reality of next-generation CRISPR. Welcome to a new frontier in biotechnology, where the story of CRISPR is being rewritten before our eyes. For years, we have celebrated this tool for its ability to edit DNA sequences with surgical precision, fixing errors that cause disease. But today, we are looking at a profound evolution: the shift from static editing to dynamic regulation. In this video, we will explore how new CRISPR variants are doubling as epigenetic regulators, offering a dual-function architecture that can simultaneously edit genomes and control gene expression. We will unpack 3 groundbreaking studies that reveal how these tools target cellular senescence and age-related diseases, moving us closer to a future where we might not just treat symptoms, but reverse the drivers of aging itself. Stay until the end, because understanding this shift from a molecular scalab to a molecular dial could change how we view human health, longevity, and the very timeline of our lives. For decades, the primary challenge in combating age-related diseases has been the irreversible nature of genetic damage and the complexity of cellular senescence. Traditional gene therapy often focused on correcting static DNA errors, fixing a broken sequence once and for all. While powerful, this approach struggled to address the dynamic, fluctuating nature of gene expression that drives conditions like cancer progression and the gradual decline of our tissues. The problem lies in the rigidity of our current tools. They are excellent at cutting DNA, but limited in their ability to modulate the ongoing conversation between our genes and our environment. When cells age, they enter a state of senescence where gene expression becomes dysregulated, leading to inflammation, loss of function, and disease. Until now, we lacked a precise mechanism to intervene in this process without permanently altering the genetic code or causing unintended off-target effects. The scientific community has been searching for a way to bridge the gap between mechanical DNA repair and functional gene modulation, seeking a method that offers both the precision of editing and the flexibility of regulation. This is where the discovery becomes truly revolutionary. Recent research has identified a new class of CRISPR/Cas9 fusions, specifically dCas9VP64 and dCas9crab, that act as a dual-purpose platform. Think of traditional CRISPR as a pair of molecular scissors that can snip out a faulty section of DNA. These new variants, however, function more like a sophisticated mixing board or a dimmer switch. By fusing the DNA binding domain of Cas9 with specific regulatory proteins like VP64 or CRAB, scientists have created tools that can bind to DNA without cutting it. Instead of making a permanent cut, dCas9VP64 acts to activate target gene transcription, essentially turning the volume up on beneficial genes. Conversely, dCas9crab induces transcriptional repression, turning the volume down on harmful genes that drive disease. This distinction is critical. It means we can now intervene in the molecular drivers of cellular senescence by reversibly modulating gene expression profiles. Rather permanently knocking out a gene, which might have unforeseen long-term consequences, these fusions allow for transient control. They utilize steric hindrance and specific molecular architectures to interact with the genome in a way that respects the delicate balance of our biology. This discovery shifts the paradigm from viewing CRISPR solely as a tool for permanent genetic modification to recognizing it as a dynamic regulator, capable of tuning the very mechanisms of aging. The implications of this shift extend far beyond the laboratory, touching the core of how we approach medicine, artificial intelligence in biology, and the human experience of aging. For biomedical researchers and clinical oncologists, this validates a new pathway for treating conditions where gene expression dysregulation is the primary driver. Imagine therapies for cancer or neurodegenerative diseases that do not rely on toxic chemicals or invasive surgery, but instead use these dual-modal interventions to restore healthy gene expression patterns. This opens the door to treating multiple age-related pathologies simultaneously by targeting the root cause, the loss of genomic stability, and the onset cellular senescence. For society, this suggests a future where the aging process itself might be reversible, or at least manageable, extending our health span without the fear of permanent genomic alteration. It challenges the assumption that gene therapy must be a one-time, irreversible event. Instead, we are moving toward dynamic gene therapy, where treatments can be adjusted over time in response to the body's needs. While we still face hurdles regarding delivery efficiency and the need for long-term safety data, the conceptual groundwork is now firmly established. We are not just fixing broken parts. We are learning how to tune the entire instrument of human biology to play in harmony for longer. Today, we have witnessed a pivotal evolution in CRISPR technology, transitioning from a static tool for editing DNA to a dynamic platform for epigenetic regulation. The research highlights that novel fusions like dCas9VP64 and dCas9crab can simultaneously edit genomes and modulate gene expression, directly addressing the molecular drivers of cellular senescence and cancer. While these theoretical frameworks promise a new era of reversible gene therapy, the journey is just beginning. The next